Thank you. So thank you very much. Thanks for having me. It's a great pleasure to be here. Is the microphone on? Everybody can hear me? Great. So today I wanted to give you a bit of an overview of the Human Brain Project, but I also wanted to give a bit of a deep dive into some of the precursor work, the Blue Brain Project. And just to be clear, the Blue Brain Project is actually, it was originally started in 2005. It's a national research infrastructure for Switzerland, and it's actually serving a role as coordinator of the Human Brain Project. But it's still continuing, and it's part of the Human Brain Project. But I'd like to start off, actually, with this poem. I went to the same high school as this woman, who wrote in 1939, I think, what is the first poem about big data. And she wrote, "Upon this gifted age, in its dark hour, rains from the sky a meteoric shower of facts. They lie unquestioned, uncombined. Wisdom enough to leach us of our ill is daily spun, but there exists no loom to weave it into fabric." And I think she really captured the challenge that many fields are facing today, but particularly in neuroscience. This is a critical problem, and the challenge is particularly big because in neuroscience we're studying many, many different types of brains. I don't know if we can dim the lights a bit. And I don't know, should I do that here? 20%? Because we're studying fruit fly brains, we're studying fish brains, studying mouse brains, rat brains, cat brains, monkey, human. And all of it, in the end, is really to understand the human brain, because that's what makes us unique, right? That's makes us who we are. That underlies our drive to create, our emotions, our creativity. At the same time, the brain is also afflicted with a broad range of disorders and diseases that we fundamentally don't understand at all. We don't know how to define them, and we don't know how to treat them. And the treatments that we have found that do help in some cases actually only work in maybe 50% of the population, and we don't understand how that works. So we're facing some significant challenge also because the pharmaceutical industry, to a large extent, is giving up on new investment into developing treatments for the brain. And part of the reason for that, part of it is the regulatory environment. Part of it is also that the leads, the information they can start from in biomedical sciences. So it turns out that 75% of the major findings that are published in Neuron and Science and Nature are not reproducible. Right? And these are the papers that are getting people tenure. So there's a big challenge there. And the fact is that it's making the field, the endeavor of developing new brain treatments, very risky, very complex, very expensive. But there's a tremendous amount of data being generated. There's even more being generated as these new brain projects emerge. You hear about them regularly. The US Brain Initiative has now targeted $4.5 billion at mapping out brain activity and brain structure and cell types and so on. So this is phenomenal. There's more and more data being produced, new techniques emerging every day to measure activity from the brain, details from the subcellular resolution, the cellular resolution, the tissue resolution, up to the whole brain scale. So more and more data is emerging every day. But the question is how do we develop a true understanding of the brain which involves linking those levels? How do we find the relationships between these different levels and across the different modalities? We need to integrate that information. So the Human Brain Project is really a 10-year initiative dedicated to triggering a global collaborative effort to drive our understanding of the human brain, to develop new methods in medicine and future computing. It's one of two PhET flagships, I'll explain what those are in a moment, that were announced last year, and it officially launched in October of last year. It currently consists of a consortium of 256 researchers, 135 research groups, 81 institutions across 22 countries. And it's earmarked for a bit over €1 billion over the next 10 years. Now, the flagships are a very interesting program because this is the first time in Europe that there was such a effort put forward, and it's not funding coming from neuroscience, it's not funding coming from health, but it's coming from the technology sector. So it's actually an unusual thing to think, what is all this technology money doing funding work in neuroscience? But it's really to drive technological innovation for the benefit of society. And so what the Human Brain Project really is it's an IT project. It's an information technology project to a large extent, building a new model of brain research, building a series of platforms that I'll describe later, but it's to drive data and knowledge integration across the various disciplines in neuroscience, and to catalyze and support a collaborative effort to understand the brain, to develop new treatments for brain diseases, and to develop new brain-like computing technologies. So these are the three key areas. In neuroscience, to integrate everything we know about the brain into computer models and simulations. In medicine, to contribute to understanding, diagnosing, and treating diseases. And in future computing, to learn principles from the brain so that we can build better computing devices and supercomputers. The Human Brain Project is divided into a number of divisions of work. It's probably too small, unfortunately, to see, but there's molecular and cellular neuroscience, cognitive neuroscience, theoretical neuroscience, brain simulation, high-performance computing, medical informatics, neuromorphic computing, and neurorobotics are the different divisions. And there is some data gathering. I don't know if I have a pointer or not, but there's some data gathering in mouse, human, and cognitive data. But the focus is on building these 6 IT-based platforms, or ICT-based platforms. And then finally, there's some work developing applications demonstrating the value of those platforms. So the data that will be generated is really strategically selected, targeted to help build the initial multi-level brain atlases needed to start building models. And that involves linking between the mouse and human brain from the level of the genome to the transcriptome, proteome, vasculature, synapses, cells, microcircuits, and so on, but identifying the similarities between mouse and human, and where do they diverge, so that you have a clear information where you can use data to derive principles that help you map, for example, between morphological or electrical properties between the two. But I really want to emphasize that the Human Brain Project is not primarily about data generation. There are many other projects that are generating wonderful data. The Human Brain Project is focused on data integration. And in fact, we're not setting out to build models because we understand the brain. In fact, we're setting out to use the process of building models to help accelerate the understanding of the brain. And that's what I'll take you through now, some of that principle. Because the idea is to build and to maintain iteratively unifying brain models. And that means models that represent the current state of our knowledge, the current state of integrated data about the brain. And that means taking data published around the world, experimental data, models from the literature, and using it to build models of circuits, neuron circuits, whole brain, run those simulations on the supercomputing infrastructure, and then analyze that, visualize that, compare it to biological data, continually validating the models and seeing how well does the integrated data actually explain the biological observations. And to do this iteratively, so that then what happens is the resulting unified brain model becomes a resource for neuroscientists to say, where does my data fit? How does it relate to other levels of activity and function structure within the brain, and how can I use that to ask questions in a more targeted fashion, more specific way. Now, we know that theory is essential to this process. It's simply not going to be possible to take a whole bunch of different measurements about the brain from different laboratories and throw them together and expect that somehow a circuit would function. Right? There needs to be organizing principles, there needs to be theories of brain function, and this is a critical and central part of the project, to have that top-down guidance, theories of how the brain is organized, combined with the bottom-up approach that integrates the details, the biological details, so that the two can meet together and basically form a connection across these levels, which has been a major challenge in neuroscience. So to do this, we're building 6 platforms. One is Neuro Informatics, brain simulation, medical informatics, high-performance computing, neuromorphic computing, and neurorobotics. And these will be open for the whole research community to use. So I'll take you through briefly some of the platforms. The Neuroinformatics platform is really there to provide the capability of accessing and organizing neuroscience data from around the world. So it provides the capability for federating data from repositories, organizing it, annotating it, and registering it to standard spatial coordinates and semantic frameworks so that it can be integrated. And then also using something called predictive neuroscience. And this is using analysis, data mining, machine learning techniques, other approaches to basically find out where can you predict the missing information. Using the measurements that do exist, how much can you predict and fill in the gaps in our data, in our knowledge? And all of the data that's going to be consumed by the brain simulation platform must be organized into this platform, into these atlases. And so that really means organizing it into species-specific spatial atlases and semantic frameworks, and also providing the ability to navigate in 3 dimensions. Where in the brain did this data come from, and what is it related to? And so some of the requirements for this infrastructure that are being planned for and implemented is the ability to share data, curate it, publish it, preserve it, federate it, search for it, make it very easy to use to deposit data, and also APIs then for accessing the data, and support for analysis and integration. And importantly, that includes tracking the provenance. So in a global collaborative project, you want to be sure that everybody gets credit for whatever their contribution is, from the level of individual pieces of data to little pieces of analysis code to major contributions. And so the way to do that is to track the provenance of every single element within the system, so that by the time you start from the raw data and you end up with a simulation, attached to that simulation output is a list of all the contributors, everybody who contributed to that outcome, to that result. This is a schematic of the neuroinformatics infrastructure, starting from a data federation. The INCF data space, the International Neuroinformatics Coordinating Facility in Stockholm, is already operating this data federation. And then on top of that, we're building sets of services for digital brain atlasing, analysis, data access, provenance tracking, and then also services for curating data privately for individual labs, and then publishing that to make it public in the global knowledge space. I'll give you a brief example of that. But all of this is designed to be sets of services that others can come in and build their own applications on top of it and extend it with other services. So the idea is that anybody should be able to build their own user portal, their own search site, their own applications that are tapping into these sets of resources. And the knowledge space, what that really is we're working with a number of different organizations, INCF, NIF, the Neuroscience Information Framework in San Diego, that already has neurolex.org, which contains a large number of neuroscience ontologies. And we're transforming that into a community encyclopedia where you'll actually have living review articles, encyclopedic summaries about each brain entity with pointers, links to the data. So you can actually go here, read a review article, as well as get pointers to the latest data, models, and literature about that entity. And behind this sits a large graph database. So this is using the latest semantic web technologies to basically make it possible for anybody, through any application, to come in and query for standardized vocabulary, but also to find data. And so that semantic search of data is really something that we see as essential, being able to find data by the producer, by the protocol, but not only one specific protocol, but related protocols. Similarly, for the specimen, brain location. Brain location can be either in terms of specific coordinates, X, Y, Z coordinates, or brain region names, which then resolve to spatial coordinates, and the data type. So we also foresee developing community ratings, other quality measures. But the process is really to go from the raw data to curate the data privately within each lab, and then to publish that when they're ready to a global public knowledge space. And just to give you an example, this is coming from the Blue Brain Project now, where they've been building an internal lab space. And this map shows you where the contributors of the data are coming from. And what it results in is a series of essentially data papers. It's probably too small in the back, but there's 6 articles here, which are basically papers describing data sets. These are data publications. They're all electronic, but they're organized very much like a standard publication. There's an abstract, there's an introduction, methods, data, and the summary. And when you get to the methods, it's actually linking out to a methods ontology. And so you can see related methods, but also you can see all the details, including the technician's manual for every step to follow to reproduce this data. So the abbreviated methods that you're used to in publications is no more. You actually directly link to the full details. Then with the data, you can basically browse the data sets by experiment, see each sample coming from the experiment, and then the extracted features. In this case, the layer thicknesses from a region of somatosensory cortex. And then the summary, because all of that information is stored in this graph database, the summary is a dynamically generated page, which basically makes a query and says, tell me the average and standard deviation thickness of each cortical layer based on the available data. And that becomes something then that's queryable for the modeler to build a circuit from. So similarly, there's data about cellular density of excitatory and inhibitory cells, molecular properties of gene expression about ion channels, morphological properties, reconstructed three-dimensional morphologies of neurons, electrophysiological properties, again, characterizing the firing properties of neurons, synaptic properties, characterizing the short-term synaptic plasticity. And then the brain simulation platform is the platform for the neuroscientists to basically make those queries to get that data and say, I now want to build a model. I want to build a model neuron using those morphologies and that electrophysiology and build up an experiment where I then test that model to see how well does it recreate other biological findings. So the idea will be brain simulation platform. Neuroscientists can log into that, build up a model, run a simulation, ask questions, instrument it with virtual instruments. So we've done things like computing the local field potential or optical dye type responses from these models, and using that to then keep validating and asking new questions from these in silico experiments. We want neuroscientists to have a tool to basically prioritize their new experiments, ask new questions. And the brain simulation platform will support a broad range of levels of modeling, from subcellular level models with particle tracking to volumetric and surface mesh and multi-compartment models and different regions, and the associated mathematics. So for example, we've got M-cell for doing subcellular reaction diffusion, We've got multi-compartment Hodgkin-Huxley models with Neuron. We've got simplified point neurons with Nest, and then mean field as well. And what I wanted to take you through now was a simulation use case. So how is it possible to use this process to actually take laboratory measurements, to take data from the lab and build a model and see, is that possible to actually build a meaningful model that is useful in understanding the biology without hand tweaking it or without having to resort to an abstraction where we've thrown out too many of the key details. So we started in 2005, the Blue Brain Project, with a set of data that had been measured over 15 years. Henry Markram's group had been measuring one single cortical circuit, the somatosensory cortex in rat at a particular developmental age, P13 to 16, all using some standardized protocols. So using standardized electrophysiological protocols to probe the firing types, again, for the synaptic protocols to map out the short-term plasticity and the synaptic response, gene expression to look at which genes for ion channels are expressed in each of these cell types, three-dimensional reconstructions of the neuron morphologies, and also for paired reconstructions to identify the putative synaptic locations between those connected cells, and as well as identifying cell types, their composition and density within this one circuit. So it's a pretty significant database all about one brain circuit. And the question is so you've done that. You spent 15 years gathering all that data. Now what do you do with it? Can you actually integrate it in a useful manner? And the challenge that we faced then was, well, to start off modeling the neurons, we had to deal with the fact that there's a tremendous diversity of ion channels that shape the electrical behavior of the cells. And this just gives you a schematic of the voltage response due to a current injection of a single neuron and all the different classes of ion channels that are actually generating and shaping that electrical behavior. So these are proteins, voltage-gated proteins embedded in the membrane of the neuron that really shape the behavior, generate the behavior. And there are about 200 voltage-gated channels in the brain. So unfortunately, if you go to the literature, there's a very, very poor characterization of any particular ion channel. In general, ion channels have not been systematically characterized under controlled conditions. So you find fragments of information that you need, and from many different species, under many different temperatures. So we started a project called the Channelone Project, which basically clones ion channels, expresses them in Chinese hamster ovarian cells, and then uses automated patch clamp robots to go in and use a standardized protocol to characterize the opening and closing kinetics of the ion channels. And that produces, the result of that is to automatically generate models of these ion channels in controlled conditions. So then, with this information, we've got the reconstructed neuron morphology, and we've got the ion channel models. We haven't characterized all 200 of them yet, but we've got representatives of each class, of the major classes of ion channels. And then we've got information with the RT-PCR data, the gene expression data, telling us which ion channels are present in those particular cell types. So the question is how do you distribute the ion channels across that neuron morphology to recreate the electrical behavior of a model neuron, of a neuron? And we used a multi-objective optimization process that basically uses as its objective characteristics, features of the firing properties of the biological cells, and is able to recreate those firing types by distributing the ion channels across the morphology. And then we tested to make sure that those generalized to other types of stimuli as well. And here we see that it generalizes very well to ramp stimulation, where it was built off of pulse stimulation inputs. But it also generalizes well to other classes of inputs, such as network-like input. And it was also interesting, we found that these models are not unique solutions, that there's a broad space of different combinations of ion channels that can produce the same firing type. And in fact, this is what's seen in biology. There is any given firing type can be created from a broad array of ion channels. In addition, we can layer in details of the dendritic activity and the dendritic processing. In this case, we used data from Jackie Schiller's lab and Matthew Larkham's lab, where they've patch clamped the dendrites to characterize things like calcium hotspots and back-propagating action potentials. And using that, then, we can layer in essentially any of these additional details into the multi-objective optimization process and automatically build model neurons that recreate those properties. So this is an extensible process. As we learn more, as we get more measurements, we can integrate them and use that to keep constraining the model further. And we recreated the diversity of firing types for both interneurons and pyramidal cells. And as well, we recreated them. We also had to have examples of the different morphological classes. So this is showing you actually overlaid many different neuron morphologies for each class of inhibitory cells on the left and excitatory cells on the right. The axon is in blue and the dendrite is in red. So you get to see these diverse shapes. This is what Ramon y Cajal called the butterflies of the soul. But these are now characterized with samples of actual reconstructed neurons. And you see there's a tremendous diversity throughout all the layers of the cortex. And then we basically know that each morphological type can embody different firing types. And so we've mapped out which firing types occur in which morphological types, so we end up with morphoelectric types of cells, and we recreated all of those. And then this is a model neuron. This is an example of one, a layer 5 pyramidal cell, which recreates then the details of the firing at the soma, but also in the dendrite of the cell. And you basically then get a fully active model with all the dendritic currents, integrating synaptic input correctly, and recreating the cellular firing responses. We also had to map out the composition of the neocortical circuit, which cell types occur in which density in each layer. And so this is kind of the recipe then for interneurons and pyramidal cells resulting from electrophysiological experiments and sampling throughout the circuit. And then we build up the model circuit using that composition to place three-dimensional morphologies in place. So I should say all of the movies after that first one are real from the model, real simulations, where now you're seeing the building process of positioning individual model neurons to fill up that space. So you see the soma being placed, and then there's this incredible tangle, very complex network with this web of axons and dendrites that's being created. And of course, the challenge is then we want to connect this circuit. It won't be a functional circuit until you wire it up and make these cells interact. But why would you expect, I mean, we're taking neurons reconstructed from different animals, and these neurons never grew up together. They didn't co-develop. So how in the world do you hope to get the neuron connections to be correct, and the synaptic positions particularly to be right? Well, sorry, one validation we did after we built that circuit was to actually do simulated immunohistochemistry for known calcium markers and proteins, calcium-binding proteins and neuropeptides, to basically compare the stains from the patterns of stains for those different classes between the in silico, the model, and the biology, and see that we were able to get the right distribution coming out of the distribution of the cell types. But back to the connectivity question. If you've got this network, we had to see what would it take to accurately predict or to get the right connections between these cells. And there are 2,970 possible synaptic pathways between the different cell types within this circuit. 22 of those have been characterized fully. And the question is if it costs $1 million to do one pathway, and it takes about a year, and we've got that many left, how are we going to do this? So the point is we need to learn principles. It's very unlikely we're going to be able to map out every single level of detail of the brain sufficiently to build a model. We have to learn the principles. We have to learn how to predict this missing information. And in this case, we had data that showed for a number of pathways the position of the synapses on the axons and the dendrites between many different classes of cells. So we checked from the model, after we just put all of the 3D cells together, we did this touch detection where we found where does the axon come close to the dendrite. And we basically said, well, there's a potential synapse at every one of those locations. What's the distribution of the locations of those synapses? And how do those locations compare to the biological locations that we've measured? And it turned out, and it completely blew us away, that for most connection pathways, the positions were the same in the model that was just randomly built as in the biology. So what it means is that the decision to actually make a synapse is actually occurring by the random apposition of axons and dendrites. Now, that's in most cases. There still are exceptions, and that was also extremely informative, because then we could say, all right, for a few types of pathways, there actually are specific mechanisms guiding the fibers in a specific way that the geometry of the neurons will not predict. And for example, the Martinotti cell creates an axon that actually follows the apical tuft, the apical trunk of the pyramidal cell, up, and it forms more synapses on the trunk than the shape of the neurons would predict. Similarly, the chandelier cells form very targeted connections to the axon initial segment. But in addition, we could layer in further information, things like the synaptic Bouton density and minimum viable connections that they need to have a certain number of synapses or not too many synapses. And as we layered in these biological observations, we found that we could predict more and more aspects, for example, the numbers of synapses per connection and the probability of connection between different classes of cells would be accurately predicted. So I think I can skip over some of this. This is going to the details of how we captured the details. So this is actually the biological measurement of the positioning of the synapses on different axonal and dendritic branch orders. And this data that we were building this off of was validated using EM. So we basically, even looking in the bright light field microscopy, you can identify putative synaptic locations that, based on the criteria that we used, are 80 to 90% accurate for real functional synapses. And then with these structural touches that I described, we ended up getting so the gray here, just to show you a little bit of the detail, the gray is where the distributions match. And then the differences in the distributions are blue and red on there. So we're seeing that, in general, we're getting a lot of the details, and then with occasional examples of where there are peaks that indicate that there is specific targeting. And then using that structural connectivity as a basis, we layer in pruning rules that, again, integrate these additional constraints on the viable connections and ultimately result in, let me go to here, a predicted connectome. So for every single cell type to every other cell type, and this is just showing the morphological types, there's a predicted connectivity there. And for the data that we have, the specific connection probabilities that we have, it matches. And then for the others, we can basically use this as a guide then for going in and performing additional experiments to map this out and validate more. Then we assigned to all of those synapses the particular class of short-term plasticity. So some cells facilitate, there's some synapses facilitate, they become stronger with use. Some depress, they become weaker with use. And that's in a very short amount of time, just in tens of milliseconds. And it's very interesting because individual neurons can actually create postsynaptic So they can form these synapses of different types. So some cells can create a facilitating synapse onto one neuron and a depressing synapse onto another neuron. And we've mapped out the rules of which types of neurons form which types of synapses, and we layer that in to build the circuit. And then when we put it all together and start a simulation, well, the first thing that we see is actually it's silent. And that's actually a relief, because I don't know how many of you build model neural networks, but usually the first thing that happens when you put it together and you turn it on is it explodes into a seizure. And here, the whole thing would be silent until we simulated something to arouse it, to awaken it up. And that would be, in this case, changing the extracellular potassium, depolarizing the cells to start making them all non-specifically active. And what happens then is this little cortical microcircuit starts to become active and produces these low-frequency oscillations spontaneously. Now, what's interesting about that is that if you do that same manipulation with a real piece of isolated cortex, it will show very similar properties in terms of these low-frequency oscillations that it generates. But then we started to instrument this with things like calculating the local field potential. And this was a tool where, because we're simulating every current source on every part of every neuron, for each individual neuron, we can calculate the extracellular potential. This is the electrical field produced by an active neuron. And then when we take all of those extracellular potentials together, we can compute the local field potential. And this is what a neuroscientist, a neurophysiologist would hear if they stuck a wire electrode into this brain circuit. You hear the neurons firing. Now, this becomes a very valuable tool because we haven't understood previously what the relationship is between the local field potential and the firing of individual cells. And in fact, the EEG, which is measured from the surface of your skull, is the same signal but filtered through a few additional layers. And we don't have a tool to understand the link between the EEG and what's actually happening in the cells underneath it. And that's what this gives us, is a tool to look at exactly that. And what that allowed us to do was to find then where, whereas traditionally it had been seen that that neurons, or the synaptic activity, was the dominating force explaining the local field potential. In fact, we see that the active properties of dendrites can have a significant impact on the shape of these signals. So this is giving us a new tool in understanding and interpreting signals that are measured from the brain. So then we've replicated that microcircuit multiple times to create a virtual brain slice. And of course, we wanted to see then, well, what's the response of this slice to stimulation? And so we modeled, we set out to recreate some experiments that had been done in slice where thalamocortical fibers to the cortex had been stimulated at different frequencies. And this is Rafael Yuste's group that basically saw if you stimulate these thalamocortical fibers at low frequencies, you get a little bit of a depolarization and then not much else happens. But when you stimulate it at higher frequencies, say 40 hertz, you get a recurrent burst of polysynaptic activity. So we set out to do that. We simulated the thalamocortical input, and here's at the 20 hertz, and I should maybe try to dim this off, dim this down. And you see not much happened. But then what we do is we go to the higher frequency, to 40 hertz, and you'll see a little bit of depolarization, this red, and then a triggering of this polysynaptic activity. And this really replicates, to a large extent, the pattern of propagation and the speed of propagation that is seen in these slice experiments with the same type of stimulation. But we know that this in vitro cortex is really not representative of what's happening in our living and working brain. right? This is all based on data of an isolated portion of cortex separated out. But then we started to simulate different conditions similar to what would happen if you were to change the neuromodulatory conditions. You were to add dopamine or serotonin and start to change the gating of, in this case, we looked at two things, calcium levels and also the activity of the potassium channels, which are also affected by acetylcholine, for example. And we looked across a broad range of conditions and saw that this circuit, so each of this is neuron activity across the different layers. And you see that sometimes it produces these low-frequency oscillations, as I had shown you before. But then as you change and decrease the calcium level and increase the depolarization, you end up in a condition that is actually recreating sort of a wakefulness-like. It's an asynchronous firing state that has different properties. And that means that this single circuit with different conditions can recreate a broad range, a whole spectrum of cortical states. And those cortical states have different response properties. So the way that they respond and compute in response to stimulation is different. In this case, in the in vitro-like condition, you provide the same stimulus, and you end up getting a large spatial response. Whereas in this other condition, this asynchronous firing, when you stimulate, you get just a focal response, linearly proportional to the amplitude of the stimulus, and then it fades away after the stimulus is removed. And we can characterize that, actually, maybe this is the figure to focus on, so we can look across these different conditions, look at the synaptic response to stimuli. But if you look at essentially the input-output function of this circuit in different states, in one state, in the in vitro-like state, it's actually recreating a sigmoid response function. So a very steep sigmoid, which basically means any input makes it very active and also makes it very spatially broad in terms of its response. In the other condition, more wakefulness-like, there's actually a linear response to the stimulus, and the spatial response is very focused, very precise. So you can see a change in the computational function of the circuit under these different conditions. So we know that we're missing a lot of things, a lot of details. And one of the projects that we have underway is to actually model glial cells and the coupling of the glia to the neurons with the vasculature. This is clearly an important aspect. We're starting just with a single neuron. It involves a lot of detailed subcellular modeling to get the pathways right for the signaling. But the goal is to model larger collections of neurons, their associated glia, and the coupling to the vasculature. And then we're also building up the infrastructure to simulate larger scale rodent-sized brains. And this is now showing the area that we've simulated in terms of those columns. We're working on building the rest. Back to the Human Brain Project, the high-performance computing platform is really the platform responsible for delivering the supercomputing power necessary to run these simulations. So they have to keep up, make sure that we've got the latest supercomputers of sufficient power. But in addition, they have to deal with problems in the way that when you start to go even larger scale with these supercomputers, you have to think about the problems that you're solving differently. In this case, we know that as these supercomputers become larger and the simulations become larger and larger, we won't be able to actually write all of the state information about a simulation off to a hard drive anymore. Because the size of that data is so big, just to fire up the hard drives will consume so much energy and cost so much that you just can't do it. So what we have to do is change the software, change the way that we're working to actually do the analysis and visualization on the supercomputer while it's running itself. So that's developing new capabilities, new software, and also new computer architectures. And we're working with, for example, IBM on that. And then this gives a bit of a map of the scale of the computation that we require, starting from a single cell model at about the gigaflop level. which is basically a reasonable laptop. It's no problem. And then we've got the first supercomputer that we started the project with was a 4-rack Blue Gene L, and that allowed us to run this single neocortical column. We've since had the Blue Gene P, which allowed us to run larger scale virtual slices, for example. And then recently we've upgraded to a Blue Gene Q, which is just a step below what's required for the cellular rat brain, but it should be capable of simulating a whole rodent, whole mouse brain. But we're, of course, in the middle of building all the software to do that. And ultimately, to run a simulation of a cellular level human brain will require an exascale supercomputer. And so this is 10 to the 18th floating point operations per second. These are big supercomputers. They're actually on the roadmap to be delivered by 2019. So this is already planned. The other industries, particularly the industry of nuclear bomb simulation, requires them. And the supercomputer manufacturers are interested to see that there are other applications. So we're very gladly talking with them and explaining our applications and making sure that we'll be able to use them as well as talking about some of the particular types of computation that are needed for these types of simulations in order to ensure that our goal is to have an exascale supercomputer capable of running the cellular level simulation by 2023. So the medical informatics platform is another part of the Human Brain Project that's really dedicated to helping drive a new way of defining disease. And this means everybody's familiar with the DSM and DSM-4 and DSM-5, which is basically the criteria to diagnose different disorders of the brain. Now, those are all based on symptomology. You go in and you make some observations of your patient and you make a classification. They have this disorder. What we want to do is we want to develop data-driven biological signatures of disease. And that means looking at actually the measurements that are coming from laboratory tests and clinical tests and brain images to help diagnose diseases in a way that we expect will be more effective at identifying potential targets for treatment than symptomology-based. So the idea is to go and use these biological samples to come up with the rules that can define diseases and ultimately look at the specific clinical features for targeting treatment. Now, the way that we're doing that is starting with clinics in Switzerland, putting in place at each hospital private mirrors of clinical data, including brain images and clinical tests and laboratory results, and making that queryable through a federation model to do large-scale aggregate analyses, so without any privacy being jeopardized, do large-scale analysis in order to develop rules and identify, well, what are the different phenotypes that make up a disease? Because we know that everybody, in this case, we looked at Alzheimer's data. All these patients were diagnosed with Alzheimer's. But we found 5 different phenotypes with specifically different relationships of changes in different combinations of brain structures. So there are at least 5 different phenotypes within this population of people diagnosed as Alzheimer's. And by using that, identifying those rules, you have a chance of being able to better address the individual variability and more specifically target a treatment. The neuromorphic computing platform is really there to provide hardware, silicon hardware, in 2 different forms for running large-scale models very rapidly and at low power. So the idea is to take the complex models. We need to learn the principles of how much of that detail do we need to keep, how much can we simplify down to retain specific functions, and then implement them in hardware. In the case of building off of the BrainScales platform, that's analog VLSI. And in this case of the Spinnaker project, that's actually using ARM cores. So these are digital chips, just like in your cell phones, and millions of them using a low-powered bus to run real-time digital simulations. And then the Neuro Robotics platform is there to provide simulated organisms, simulated sensory input, and simulated motor output in a virtual environment so that you can perform closed-loop simulations that really ensure that you can capture and model behavior with the model circuits. And of course, some of the potential applications of using these platforms are to investigate the principles of cognition and use a virtual mouse, for example, in a virtual maze, looking at visual stimuli. And when it makes a decision, you can trace back through every piece of activity, through every pathway, what was it that resulted in that decision? What were the mechanisms that actually generated that, or that memory retrieval? You can use it to identify potential targets for drugs, and also identify which targets would have possibly negative side effects. And also to potentially create new types of controllers for both industry and consumer devices, using brain-like principles. There's also a number of important activities here. In ethics, ethics in society is actually a major portion of the project. 3% of the budget goes to that, to make sure that there's a continuous, ongoing dialogue with the public about what's happening within the project, what are the developments, what are the concerns, listening to help shape the direction and the policy of the project as the science emerges. Also, we need to be continually training the next generation of scientists with these multidisciplinary skills to be able, and transdisciplinary skills to come in and really work on such a project. Of course, dissemination is key, making sure that the awareness of what we're generating, and also open access policy is standard for publications, data, and tools. And then governance of this. It's a very large organization that we really aim to have closely working with other major international efforts, and that is a significant part of the project. So you've got an organogram here, which I think is trying to show itself. And it's too small to be seen, but I encourage you to go to the website to look at it in greater detail. But the key point is, we don't want this organogram to remain static over 10 years. It's really essential that we have a continual influx of the best people, best ideas, best technologies continuing. And what we've done in the first few years is already had, in the first year actually, is to have an open call where 20% of the budget was put into an open call for additional partners to come in and contribute new elements to the project. In the next phase as well, we'll have further efforts with even more of the budget coming from partnering projects. So thank you very much for your attention. I'd be glad to answer any questions. Thanks.
Have we got any questions? There's a microphone that'll come to you.
Hello. Yeah, you said so yourself that it's remarkably difficult to recreate experiments, neuroscience experiments that others have done. It's very difficult. So going forward, how do you imagine your validation process, especially on the macroscopic scale and on the global scale? How do you imagine that will work out for you?
So I think the key is to really having the detailed methods. And very often this is lacking. And collaboration is one way now if you really want to recreate results. That's the best chance you have of recreating, is having a collaboration with individuals. We need to promote, though, a new publishing model that simply contains the original data, that contains the detailed methods, that you can actually have a chance of recreating the analysis results so that you can see at each level all the details. Because right now what happens is you end up seeing the idealized summary figure that shows just the point that the person wants to make in their publication, and you miss out on all of the rest of the details in many cases. So we want to really have a new publishing model that exposes and makes the results themselves more reproducible.
I think we have another question in the middle.
Yeah. I hope this isn't too on tangent to what you're talking about, but are you working with scientists on brain-machine interface type problems? And if so, how does your research relate to that?
So it's a very good question. Right now, we don't have a component on brain-machine interfaces in the project. I'm sure it will come. Within the Blue Brain project, we are doing work in that area, and we've done simulations of multi-electrode arrays and interfacing that to tissue in terms, and also being able to record from those simulated electrode arrays, but also use them for stimulation and see which neuron populations are activated and neuron fibers are activated. We have other work that's under development for other types of stimulation as well. So it's an active area of interest, but it's early days.
There's one up here.
So my question is in regards to translation of the knowledge base, the huge knowledge base that'll come from it into a clinicians. You know, when I sit in clinic, for instance, how is that going to translate to the children whom I see? And there I unfortunately can see lots of loops, lots of problems, and maybe not necessarily, or certainly not in the next, I don't know, decade or two, although that might sound cynical, something that'll actually tell me at the time I'm seeing a child, because a lot of the data you've talked about is on the adult brain, which may or may not necessarily translate into the children's brain, depending on the ages, into how I'm going to alter the management, for instance, of a child with epilepsy based on the data.
So I focused on one particular use case because I had a lot that I could show for that. But the idea is that this, in fact, we're talking now to other large-scale clinical projects to take their data and curate it into the same platform. So that, for example, there are projects that are capturing animal model measures from animal models as well as clinical environments. And what we want to do is make sure that that data gets curated into a common vocabulary and a common ontology so that you can actually find relationships between data that's coming from basic research and data that's coming from the clinical environment in order to see those relationships that we simply can't see right now. So there's a significant effort that will be put into curating many different types of data from traumatic brain injury, epilepsy, probably MS, into a common vocabulary and with a way of identifying, for example, which data is showing you responses to inflammatory or inflammatory responses, but in a way that's independent of the disease model, so that you can look at common mechanisms without the bias of the particular disease being studied. So this is the goal. We've got a number of different projects that we're really aiming to work with to curate it, but it's clearly going to take a lot of time and a lot of effort to do it.
Thanks.
Yep.
Regarding comparative neurobiology, which of your groups and which other external institutes or journals or websites look at the difference between human and other mammal and primate brain structure and chemistry? And for example, what do you think would be some of the main differences suggested that it's maybe less the neurostructure and more the percentage of white matter of connections?
So actually one of our collaborators, Javier de Felipe, has done a lot of studies of the differential anatomy between species. There's also work at the Allen Institute that shows gene expression in mouse versus human. I don't have an answer for you about what the key differences are, but what we see is that there are elements at each level that are very similar, and then there's divergence. And basically that point of divergence is what really interests us, so we can see how much can we predict of the human aspect, and where is it we're simply going to have to get additional information.
So I think we have time for one more, if anyone's... There's one at the back.
Thanks very much. That was a really interesting lecture. It seems like you're approaching the problem from a sort of bottom-up reverse engineering of the brain. What about a more top-down system engineering approach where you start off with behavior and finish up at neurons rather than the other way around?
Right. So actually, that's one of the things I mentioned, but I couldn't show you today. But absolutely, I think both approaches are essential. And the whole infrastructure is being built to support both directions, because we want very much that these high-level behavioral and cognitive models come in, and we have the bottom-up as well, and that the two of them can be reconciled to identify where can we actually bridge between those levels. So both are essential and part of the approach.
So thank you all for coming, and thank you to London Technology Week, which was promoted through. I know quite a few of you came in from outside of Imperial. And our next big speaker talk will be on October the 1st, where we're hosting Usama Fayed, who is Chief Data Officer of Barclays and formerly the first Chief Data Officer of Yahoo. So please do come along for that and sign up to our mailing list on the Data Science Imperial website. If you Google us, you'll find us. And of course, a big thank you again to Sean Hill.